this, we need to understand what are essential and what are non-essential amino acids. We know what the definition. I don't want to go in detail what is essential and what is non-essential, but here this names of these essential and non-essential amino acids which are synthesized in our body, which are not synthesized in our body. At least if you can remember these names for non-essential and essential. But there are some amino acids which we consider as conditionally essential amino acids. So those amino acids also we have to remember. When you say essential, starting from valine, all these side chain amino acids, then phenylalanine, aromatic amino acids, tryptophan, then hydroxy amino acid, threonine, then methionine, lysine, basic amino acid, histidine, all these are essential amino acids. And similarly here, asparagine, aspartic acid, glutamic acid, so all these which we consider as non-essential amino acids. So here this classification is mainly based on function of the amino acids, based on the function. The previous classification is based on nature of the organoid. So this is not important, but then we need to understand at least example for essential and non-essential. That's why I have added this particular slide here. Next one is metabolism of amino acid. Metabolism of amino acid. Metabolism of amino acid. How amino acids are metabolized in our body. How it is metabolized in our body. How it is being utilized in our body. That is metabolism of amino acids. We understand the metabolism of carbohydrates. Various metabolic pathways are there and disorders which are related to the carbohydrate metabolism. In the same way, even amino acids are also metabolized. So with the following mechanism. So here definitions are very important. General mechanism, general reactions are very important. If you are not able to, if you are not confident with specific reactions, at least you can remember the general reactions. General reaction is very important. Definition is very important. Transamination, deamination and decarboxylation. To begin with transamination, transferring amino group from amino acid to keto acid, so that it results with formation of new amino acid and a new keto acid. So that reaction is called as transamination. So this is one amino acid and this is one keto acid. So when these amino and keto acid reacts with each other, so which is catalyzed by transaminase, it results with formation of new amino acid and a new keto acid. So this is new amino acid. You just specify what is the amino acid formed here. For example, if you consider R1, R2 or R1, R2, you write R3, R4. Okay, so that general mechanism is very important. Transaminase. Specific example we can write alanine transaminase, aspartate transaminase. Alanine transaminase. Alanine is the starting substrate. Alanine we know it's a neutral amino acid. Amino group is there, carboxylic group is there, additional methyl group is there. That is how we can easily remember the structure of alanine. And this is amino acid and this is keto acid. Keto acid. This keto acid also we can easily remember because if you see this structure, it is almost similar to the structure of glutamic acid except this position, right? Except the keto group. So if you remember the structure, so because it's a keto acid, right? So see a keto group is exists. So alpha ketoglutarate. Again we know already we discussed about alpha ketoglutarate because alpha ketoglutarate is mainly involved in TCA cycle. It's a major intermediate of TCA cycle. So alanine amino acid reacts with keto acid. You just remember here the this keto group is replaced by amino group and this amino group is replaced by keto group. That is how we get this structure exactly. So here we get pyruvate. Pyruvate we know already we discussed about pyruvate, how glucose is oxidized to pyruvate. And here glutamate at this position CH and NH3, right? Amino group is added. So when amino acid reacts with keto acid, alanine reacts with alpha ketoglutarate. That means so it forms new amino acid, glutamate, and new keto acid is pyruvate. And what is the enzyme here? Alanine aminotransferase. Alanine aminotransferase. Okay, ALT. Another example is AST. Okay, so this one is AST. AST, aspartate is the starting substrate. Similarly, oxaloacetate, Oxaloacetate is a keto acid and an intermediate of TCA, and the enzyme is aspartate transaminase. So here it results with formation of new amino acid again and new keto acid. So these two examples, please remember, this is very important. We need to understand what is definition for transamination. So what is the role of ALT as well as